How many times in your life have you taken antibiotics? Chances are you've lost count. When the first antibiotic was discovered in 1928, it transformed bacterial infections from a death sentence to a mere inconvenience. But almost 100 years later, the misuse of these drugs has led to bacteria evolving immunity against them. In 2019, more than 1 million people died from an antibiotic-resistant infection, and the problem is only growing. Enter bacteria's natural predator, the bacteriophage, or phage for short. This is a tiny, simple virus, which sometimes looks a bit like a spider. While you might think of viruses as our enemy, phages are our friends. They only attack bacteria. Now, this sounds like the perfect solution to our problem, but phages have two key limitations. First, they're hyperspecific. Each type of phage can only infect a certain subgroup within a species of bacteria. Second, not all phages kill their bacterial host. After infection, more copies of the virus are made inside the cell. Some phages burst out, killing their host, but other types leak out, keeping the bacteria alive. My goal is to overcome these barriers by making genetic and chemical modifications to phages. My first task was to increase the variety of bacteria we can target. I started with a bacteriophage specific to the species E. coli, which you may be familiar with if you've ever eaten at Chipotle. Now, this phage uses its spider-like leg proteins to bind a thin, wiggly appendage called a pili, present on some, but not all types of E. coli. By editing the phage's DNA, I can alter the expression of the leg proteins so they can bind other targets, such as a specific carbohydrate found on over 70 different microbes. My experiments confirmed that the genetically modified phage can now bind E. coli lacking this pili, along with entirely different species. Now, normally, this phage does not kill its E. coli host, so my second task is to turn it into a tiny assassin with the help of synthetic polymers. Chemists make polymers by linking together repeating units of smaller molecules to form a long chain, much like beads on a string. Certain polymers are attracted to the surface of bacteria, killing them by disrupting this outer layer. Some of these polymers are so potent bacteria cannot develop resistance to them, but they're also harmful to our own cells. My approach is to chemically link these polymers to the exterior proteins of the phage not involved in binding bacteria. Thus, the genetically modified phage acts as a shuttle to direct the polymers towards bacteria and away from human cells. By combining genetic and chemical engineering, we can create novel phage-based therapies to pick up the fight against bacteria where antibiotics left off. Thank you.